Dr. Sage here. Today we're going to discuss Chapter 17. Chapter 17 is where we talk about how the DNA is actually used. And this is overall known as the central dogma of molecular biology. Basically, what happens is information is stored in the DNA. That DNA is transcribed into RNA. And then that RNA is translated into protein. So this is the overall flow of information. Information flows from DNA to RNA to protein, and that's known as the central dogma of molecular biology. So what that means is what your DNA is actually doing, the information that it's storing, is that information is telling you how to build proteins. So RNA is a bridge between genes and the proteins that the genes code for. Transcription is the synthesis of RNA under the direction of DNA. Transcription produces a particular type of RNA called messenger RNA or mRNA. We're actually going to talk about three different types of RNA today: mRNA, tRNA, and rRNA. There are actually more than three types of RNA, but to learn about the other types, you need to take a higher level biology class. And then translation is how you synthesize a polypeptide or a protein using the information that's stored in the mRNA. And ribosomes are the places where you're going to do translation. We've talked about them in prior chapters. They're basically the workbenches where you build your protein. So we'll talk about them again today. Okay, so this happens a little bit differently in eukaryotes versus prokaryotes. So in a prokaryote, like a bacteria, okay, they have DNA, and then they take their DNA and they do the process of transcription to make mRNA. And then that mRNA goes to a ribosome, and the ribosome reads the information in the mRNA and uses that information to build the polypeptide or the protein. And that step is called translation. So they do transcription, DNA to mRNA, and translation, mRNA to protein. Now, eukaryotes were a little bit different. For one reason, remember, our DNA is stored inside our nucleus. So we're going to do transcription. That's where we take the information in the DNA and copy over that information into RNA. Now, of note, when we copy over the information, we do not copy the entire piece of DNA. We don't copy the whole chromosome. Okay, because what we need is we need more of a particular protein. Like, let's say that right now we need more insulin, which is a protein. What's going to happen is our cell is going to go find the chromosome that insulin is on, and then it's going to copy the piece of DNA that codes for the insulin protein. In other words, copy the insulin gene from DNA over into RNA. So it won't copy that whole chromosome that insulin's on, it will just copy that one gene. Okay. Now, another thing that's different besides the fact that we have a nucleus and prokaryotes don't is our mRNA, when we make it, it's not actually ready to be used. So it's called pre-mRNA, or the primary mRNA transcript. And then we have to do a process called RNA processing to get the mRNA actually ready to be used, get the final mRNA transcript. Then the mRNA leaves the nucleus and goes into the cytoplasm. Okay, it finds a ribosome. The ribosome reads the information on the mRNA, uses that information to build a polypeptide or protein, and that step is called translation. So in eukaryotes, we have transcription, DNA to pre-mRNA, that's transcription. Then we take the pre-mRNA, do RNA processing to make the final mRNA transcript. Then the mRNA is read through the process of translation to build a polypeptide or protein. Okay, so this is kind of like the big picture of what we're going to be learning today. And we will learn the details. We'll learn what happens during transcription, during RNA processing, and during translation. But this is a big picture, the overview of what we're going to be learning today. Now, your DNA stores information. And the information it's storing is telling you how to build proteins. And that information is stored in a code, and the code is actually in the nucleotides, and in particular the base pairs. Those letters A, T, C, G, they're used as a code to tell you how to build your proteins. And remember that proteins are built out of amino acids. Recall from the macromolecules chapter that there's 20 different amino acids. Okay, so what's going to happen is these letters of your genetic code are going to be used to form words. And the words are going to tell you which amino acid to be using to build the protein you're trying to build. And it turns out that the words that are going to be formed to tell you which of the 20 amino acids to use are all 3-letter long words. Okay? And those 3-letter long words are called codons. So let me do that in an example. So let's say that you need more of your insulin protein right now. What's going to happen is you're going to find the chromosome that the insulin gene is on. You're going to find that particular gene. Let's say this is the insulin gene right here. That insulin gene is made up of a sequence of base pairs, letters, A, T, C, G, that codes for that insulin gene. So you're going to take that DNA and you're going to do transcription to make an mRNA copy of that DNA. Now, transcription is based on complementary base pairing. Recall that in DNA, A pairs with T and C pairs with G. Also recall that RNA does not have the base T. In place of the base T, it has the base U. So what we're going to do is we're going to make an mRNA copy of that DNA gene based on complementary base pairing. So A would pair with U, C with G, A with U, C with G, G with C, etc. So you're going to make this mRNA copy of this piece of DNA. Okay, now this mRNA is a code that's going to tell you how to build the protein you're trying to build. The process of going from mRNA to protein, that process is called translation. Now the code that tells you which amino acid to use, those are 3-letter long words called codons. For example, the code UGG tells you to use the amino acid tryptophan, UUU phenylalanine, GGC glycine, UCA serine. So it's telling you how to build your protein. Because remember, a protein is just a sequence of amino acids. Which amino acids you use and the order you use them in determines which protein you're building. How do you know how to do that? Well, that's based on the 3-letter long words, the codons in the mRNA, which, how do you know that? That comes from the DNA. Okay, so that's kind of the big picture of what's going to be happening. And that shows you these 3-letter long words, codons. Now, since there are 4 different letter choices, A, T, C, G, and there's 3 different spots that you can put them in because every codon is a 3-letter long word. So there's 4 choices for each of these 3 different positions. What that means is there's a total of 64 different codons that can be made. Okay, so there's 64 different codons you can possibly make. And that makes up the genetic code. So, for example, this is the genetic code. This is telling you like which codons specify which amino acids. So, for example, UUG says use the amino acid leucine, whereas UCC says use the amino acid serine. So that's the genetic code. Now, of note, there are 64 different codons, but there are only 20 different amino acids. So because of that, the genetic code is redundant. What that means is more than one codon can code for the same amino acid. So for example, all 6 of these codons from UUA to CUG, they all code for the same amino acid. They all code for leucine. So the genetic code is redundant. But the genetic code is not ambiguous. What that means is, for example, AAC always codes for asparagine. AAC would never code for glutamine. It would never code for lysine. AAC always codes for asparagine. So the genetic code is not ambiguous. So the genetic code is redundant. AAU and AAC code for asparagine, but it is not ambiguous. AAC would never code for lysine or glutamine or histidine. Something else to note about the genetic code is there are a couple of special type of codons. Okay, these are called the start codon and the stop codons. Okay, so the start codon, what that does is that says start translation here. In other words, start building your protein. Now, for this class, for Bio 1, you don't have to memorize all the different codons and all the different amino acids they code for. Stay in science like take biochemistry, you'll probably have to memorize that. But there is one you might need to memorize, okay, and that's the start codon. So AUG is the start codon. It says start translation here, start building the protein. And it does also code for an amino acid. It codes for methionine. Okay, so that's the start codon. Now, there are also stop codons. Okay, now you do not have to memorize their letters. Okay, like, UGA. You don't have to memorize that. You should just know that there are stop codons. That the stop codons say stop translation here, stop building the protein. And also that the stop codons do not code for any amino acid. Okay, so those are the start and stop codons. And overall, these are all of the codons. And the codons are 3-letter long words that tell you which amino acid to use to build the protein you're trying to build. Okay, and this is kind of very similar to how, we have 26 different letters in our alphabet, okay? And which order you place those letters in, so which letters you use and what order you use them in, determines the words that you use to spell out sentences. That's very similar to how we have these, 4 different letters, A, T, C, G in DNA, or A, U, C, G in RNA. And those 4 letters used to form 3-letter long words. And those 3-letter long words code for the different amino acids, different words. Okay, but if you are writing a sentence, how you know where one word ends and the next word begins is because you put a space between them. Okay, well, there are no spaces in your DNA or your RNA. So how do you know how to read the words? So, for example, let's say that I write a bunch of letters up here, but I don't put any spaces between anything. Okay, so there's no spaces. But let's say I tell you what I want you to do is I want you to read the first 3 letters, and then read the next 3 letters as a word, and then read the next 3 letters as a word. Okay, if you do that, then you can read the words dog ate cat, even though there's no spaces between those letters. Okay, and that's how the mRNA is read. That's called reading within the reading frame. Okay, so you read 3 letters, and then you read the next 3, and then you read the next 3. That's reading within the reading frame. Now, there might actually be letters before that or after that. So let's say we have these letters. Okay. So, how do you know where to start reading? Okay, let's say I tell you, what I want you to do is the very first time you find the letters DOG, I want you to start reading. So I'm not going to read this as Todd. Okay, what I'm going to do is I'm going to start reading the first time I find those letters DOG. Okay, that would be equivalent to the start codon. That's where you start reading the reading frame. And then I say what I want you to do is I want you to keep reading 3 letters at a time until you find the letters CAT. Okay, so I'm going to read dog because that's where I'm going to start reading. That's the start codon. And then I read 8, and then I read cat. And now I'm going to stop reading. So cat is equivalent to the stop codon. So even if there's letters before it or after it, you don't read those letters because you start at the start codon, you read 3 letters at a time until you reach the stop codon, and then you stop reading. So that's how you actually read the genetic code, the mRNA. Now, another thing to note is that the genetic code is not just for humans. Like, this is not the genetic code of humans. This is the genetic code of basically everything that's alive. We all read the same genetic code. And it didn't necessarily have to be that way. Because these are words that mean things. You know, like AUG means leucine, CCC means proline. And it's kind of equivalent to how I use the letters DOG to mean that animal that I call a dog. Or I use the letter CAT to refer to that animal that I call a cat. But the only reason I'm using the letters DOG is because I happen to be speaking English. If I wasn't speaking English, or if I was speaking a different language like German or Spanish or French, I might use a different set of letters to mean the same creature that I call a dog. Okay, so we as humans, we actually speak different languages. We use different letters to mean the same thing, like DOG to mean dog versus a different set of letters to mean that same creature that I call a dog. Okay, so we as humans speak different languages. But it turns out all living things speak the same genetic language. Okay, we all use the same genetic code. And it did not have to be that way. Just like humans speak different languages, different organisms, hypothetically, could speak a different genetic code. The reason we all do speak the same genetic code has to do with descent from a common ancestor and evolution, which that's not the focus of Bio 1, but that's why we all speak the same genetic code. But the fact that we all speak the same genetic code allows us to use other organisms to help humans and to better understand humans. So, for example, some humans, we've been talking about insulin as an example, some humans need to take insulin injections. Okay, insulin is a protein that humans make. Okay, so how do you get human insulin protein to give to a human? You don't take it from one human to give to another human. You wouldn't be able to make enough insulin that way. So what was done instead is they take the human insulin gene. So they take the piece of DNA that codes for insulin out of a human, out of a human skin cell. They take that piece of DNA that codes for the human insulin gene, and they put that DNA in some other organism, like a bacteria or a yeast cell. Now, because yeast have the same genetic code as humans, what happens is when you put that human piece of DNA into a yeast cell, what the yeast are going to do is they're going to build human insulin protein. Okay, so what we do is we use these organisms like bacteria, yeast as like little factories for us to grow up a bunch of human insulin protein. And that's how we get enough human insulin to give to humans who need to take insulin injections. So because we speak the same genetic code, we can use this genetic code to help humans. Like, for example, make human proteins in little factories of bacteria or yeast to get the proteins that we need. We also use it, besides for medicine, like getting insulin, we use it for science. There's a lot of experiments that are unethical or unfeasible to do in humans. Okay, but let's say you want to study some human gene and you can't do the experiment you want to do in a human. Well, put that gene into a yeast cell or a fruit fly, and then you can study that gene in yeast or fruit flies. Okay, and since they're going to build the same protein as we would build, we can learn about that protein in that gene. Okay, so, and here's an example of that. So, for example, you can take that little organism that you call a lightning bug or a firefly. You know, they can glow. The reason they can glow is because they have a gene that codes for a protein called luciferase. We'll take that gene out of a firefly and put it into a plant, and then you'll have a plant that will glow just like a lightning bug. Or jellyfish can glow. The reason they can glow is because they have a protein called GFP that emits light that glows. Okay, we'll take the gene for GFP, green fluorescent protein, and put that gene into a pig, and then you'll have a pig that will glow just like a jellyfish. Now, honestly, why they did this, I have no idea, because we don't do this just for fun. We do this to help humans. We do this for medicine or for science, is why we make these transgenic organisms. But there is actually one we do have just for fun. Like, you can't go out and buy this or buy this today, but run down to like Petco or Pet Supermarket, go to the aquarium section and find the fish called glowfish. Okay, glowfish, a little fish that glow, they emit light. But that species of fish does not naturally emit light. And we're not injecting glowing dyes in those fish to make them glow. What happened is they took the gene for green fluorescent protein from jellyfish, and they put that gene into those fish. Now those fish have those genes, and those fish will make the green fluorescent protein just like the jellyfish do, and now those fish will glow. Okay, and that's how you get those glowfish. So they are a transgenic organism. Okay, so that was 17.1, which is kind of the big picture overview of what we're going to be learning today, which is transcription, DNA to RNA, RNA processing, and then translation, RNA to protein. Now we're going to start going into some of the details, starting with transcription. Recall that transcription is how you take the information in DNA and copy that over into mRNA.
The first step in protein synthesis is transcription of RNA from DNA. The portion of the DNA that is transcribed into an RNA molecule is called a transcription unit. An enzyme called RNA polymerase carries out transcription. With the help of protein transcription factors, it attaches to the beginning of a region of the DNA called the promoter, pries the DNA strands apart, and untwists a short portion of the double helix. RNA polymerase moves along the DNA, pairing up RNA nucleotides with their DNA complements, adding nucleotides to the end of the growing RNA molecule. Here's a close-up view of elongation. Only one DNA strand, the template strand, serves as a template for RNA synthesis. RNA polymerase moves along the DNA strand in the 3' to 5' direction, adding nucleotides to the 3' end of the RNA chain. Note that U in RNA pairs with A in DNA.
Once transcription is complete, RNA polymerase releases the completed RNA and detaches from the DNA.
Okay, so remember, transcription is how you take the information that's in the DNA and copy that information over into mRNA. Now, in order to do that, you need an enzyme that can make an RNA copy of the DNA. And there is an enzyme, and honestly, that enzyme should be fairly easy for you to remember. Because recall in the last chapter, when we talked about DNA replication, we talked about an enzyme that makes a DNA copy of DNA, and that enzyme is called DNA polymerase. Okay, well, the enzyme that makes an RNA copy of DNA has the same name. It's just called RNA polymerase instead of DNA polymerase. So RNA polymerase is the enzyme that makes the RNA copy of the DNA. Another thing to note is a difference between DNA replication and RNA transcription. When DNA polymerase makes the DNA copy of the DNA, it copies the entire piece of DNA, the entire chromosome. When RNA polymerase makes the RNA copy of the DNA, it does not copy the entire piece of DNA. It does not copy the entire chromosome. It copies one small part, which is one gene. Okay, now there's a lot more details about transcription. Obviously, there's a set of enzymes involved. There's a process to do transcription. But we're not going to learn those details for this exam. So that's the extent that you need to know for transcription. Okay, recall that one difference between eukaryotes and prokaryotes is when you make the RNA through the process of transcription. In eukaryotes, the RNA is not ready to be used as soon as you make it. You have to do an extra step called RNA processing. So we're going to learn what happens during RNA processing. what's going to happen is that pre-mRNA that you're making, you're going to change both ends of that pre-mRNA molecule. Remember, RNA has two different ends. It has a 5' end and a 3' end, just like DNA does. Okay, well, we're going to change the 5' end and we're going to change the 3' end. Okay, this is 2 out of the 3 things that's going to happen during RNA processing. So the first 2 things we're going to talk about that happen during RNA processing is on the 5' end of that pre-mRNA molecule, we're going to add a 5' cap. Okay, and what the 5' cap is a modified G base. You know, the base is ATCG or AUCG in RNA. Okay, it's a G that's been chemically modified a little bit. Don't worry about for this course how it's been chemically modified. Okay, so that's called the 5' cap. That's we're going to add to the 5' end of that pre-mRNA molecule. Then on the 3' end, we're going to add what's called a poly A tail. Okay, recall poly means many, and A is referring to the A base. So what we're going to do is add 200 A bases onto the end of this RNA molecule. So those are 2 out of the 3 things you do during RNA processing. Now before we talk about the third, let's talk about why you do these 2. Okay, well, these 2 things, the 5' cap and poly A tail, one thing they do is they help the mRNA to leave the nucleus and go to the cytoplasm. Another thing they do is they help the mRNA to attach to ribosomes, which is where they're going to need to go, so the ribosomes can read the mRNA to make the polypeptide or protein. The other thing they do is they protect the mRNA from hydrolytic enzymes. Okay, what that means is your cells make mRNA. Your cells also make an enzyme, this little Pac-Man here, whose job it is to destroy mRNA, to go along and chop it up, to eat it basically. Okay, so your cells make mRNA and they make an enzyme that destroys mRNA, but then they add a 5' cap and a poly A tail, which will prevent this enzyme from destroying the mRNA. Okay, so that's the purpose of the 5' cap and the poly A tail, is to protect your mRNA from your hydrolytic enzymes. All right, so here's an example of a gene inside a eukaryote. Okay, and I'm going to explain this in order to explain the third thing that happens during RNA processing, which the third thing that happens during RNA processing is called RNA splicing. Okay, so this is one gene that's displayed here in this DNA. You know it's one gene because the gene starts at the promoter sequence and the gene ends at the terminator sequence. So between the two, that's one gene. Okay, between those two, you also have the start codon. Remember, it says start translation here. Stop codon, it says stop translation here. So basically, between the start codon and the stop codon, that chunk of DNA, that should code for the protein that you're trying to build. So what happens is you start at the promoter and go through the terminator and copy this DNA into pre-mRNA. But it turns out that there are pieces of DNA that are going to code for the protein that you want to use. Those are called exons. Okay, but there are also pieces of DNA that do not code for the protein that you want to build. Those are called introns. So when we make the pre-mRNA, it has both the exons that you want and the introns that you do not want. So if you were to start at this start codon and read through this pre-mRNA to the stop codon and build your protein, you would build the wrong protein. Okay, that protein would not work. The reason it would not work is because you have chunks of mRNA that are not coding for the protein you want to build, the introns. Okay, so what you need to do is you need to do RNA splicing. Splicing is where you cut out and remove the introns. You get rid of the introns, and then you paste together the exons to make the final mRNA transcript. Now, if you start at the start codon and read through to the stop codon, when you build the protein, you will build the correct protein. Okay, so exons do code for the protein you want to build. Introns do not code for the protein you want to build. What you need to do is you need to cut out and get rid of the introns and paste or splice together the exons. That's called RNA splicing, which is the third type of RNA processing. Recall that RNA processing only happens in eukaryotes. So the 5' cap, the poly A tail, and RNA splicing, this is only done in eukaryotes. It is not done in prokaryotes. Okay, for example, prokaryotes don't have introns. Only eukaryotes have introns. So RNA processing is only done in eukaryotes. It is not done in prokaryotes. Okay, the next topic is, for now, just a definition to memorize, and you'll see why this is important in a few minutes. Okay, so we've learned a lot about enzymes. Enzymes are proteins that are a catalyst. In other words, a protein that will speed up a chemical reaction. Okay, well, it turns out that enzymes are not the only catalyst that your cells use. Your cells can also use a different type of catalyst called a ribozyme. So a ribozyme is an RNA molecule that is a catalyst. In other words, an RNA molecule that will speed up a chemical reaction. So another way of thinking about it is a ribozyme is an RNA molecule that behaves like an enzyme. It's not an enzyme because an enzyme is a protein and a ribozyme is a piece of RNA. But a ribozyme is an RNA molecule that behaves like an enzyme. Okay? In other words, it's an RNA molecule that is a catalyst, an RNA molecule that will speed up a chemical reaction. Okay, and you'll learn a little bit why ribozymes are important. The next section of the chapter talks about translation. Remember, translation is how you take the information that's in mRNA and use that information to build your protein. Okay, so we're going to learn about the process of translation now. And we are not going to cut all the details of it like I did with transcription. So in order to do translation, what you need something that can read the information that's in the mRNA and convert that information over into proteins, which is a different type of macromolecule. Okay, what's going to do that is another type of RNA called tRNA, or transfer RNA. Okay, what the tRNA is going to do is it's going to read the codon on the mRNA and then bring the correct amino acid for that codon. Okay, so let me explain it here. So here's an example of a tRNA molecule. So this is one piece of RNA. If you go from the 5' end and follow through to the 3' end, you'll see that's one piece of RNA. But the RNA does fold and base pair with itself. You know, C pairs with G, A pairs with U to form this structure here that we're seeing. So what's going to happen is, let's say we have a piece of mRNA down here. That mRNA has 3-letter long words called codons. Like, for example, let's say UUC. Well, there's a spot on the tRNA down here called the anticodon. Okay, what the anticodon is going to do is it's going to complementary base pair to the codon. So GAA would base pair with UUC. A pairs with U, G pairs with C. Okay, so the anticodon is on the tRNA, whereas the codon is on the mRNA. The anticodon on the tRNA complementary base pairs with the codon on the mRNA. So this one would be UUC, for example. Okay, in this kind of cloverleaf structure here of the tRNA, the 3 bases down here at the bottom, that is always the anticodon, even if it's not labeled. The 3 bases down here will always be the anticodon. Now that anticodon codes for a particular amino acid. Okay, whichever amino acid it codes for, that amino acid is going to be attached to the tRNA up here. Okay, and only that amino acid is going to be attached. Okay, and that's how you read the genetic code. Because remember, the codon codes for a particular amino acid. Okay, well, whatever amino acid UUC codes for, that amino acid and only that amino acid will be attached to this tRNA molecule. So what that's going to do is it's going to read the information in the mRNA and use that information to bring the correct amino acid. And that's how a tRNA molecule works. Now, a little more realistically, it would look like this. So the anticodon would be down here, and the amino acid would be attached up here. And the rest of the figures, it's going to be diagrammed like this, just so it's easier to visualize. So the anticodon is down here. You would attach the amino acid up here on this tRNA molecule. Now, in order to correctly do translation, to correctly build your protein, a couple of things need to happen. One thing that needs to happen is you need to correctly match up the anticodon on the tRNA with the codon on the mRNA. Okay, so how that's going to happen, the correct match, that's going to happen inside the ribosome, and we'll talk about that in a minute. But the other thing that needs to correctly happen is you need to make sure that the correct amino acid is attached to this tRNA molecule. Because there's only one amino acid that should be attached to this one, whatever amino acid the codon UUC codes for. And only that amino acid. Okay, and there's an enzyme that will put the correct amino acid on the correct tRNA, and that's called aminoacyltRNA synthase. Okay, so that is the enzyme that puts the correct amino acid on the correct tRNA. So what this means is you have multiple different tRNA molecules because you have different amino acids. Remember, you have 20 different amino acids, 64 different codons that code for those 20 amino acids. Okay, so you need to have different tRNAs because you're going to have different anticodons on the different tRNAs that match up with the different amino acids. But it turns out you do not actually have 64 different tRNA molecules. So to save your cells some time and energy, what happens is some tRNA molecules will match up with more than one codon. Remember, the genetic code is redundant, okay, which means more than one codon can code for the same amino acid. A lot of time that redundancy is in the third base. So for example, UUC and UUG, they might code for the same amino acid. So a lot of times the third base in the codon is not as important, and there's a word for that, and it's a funny word, it's called wobble. So wobble refers to the fact that oftentimes the third base in the codon is not as important, and that's what accounts for a lot of the redundancy in the genetic code. Okay, so remember that ribosomes are the workbenches where you're going to build your proteins. So protein synthesis, or building proteins, that happens on the ribosomes. And ribosomes themselves are built out of two different things. Ribosomes are built out of proteins, and they're built out of a particular type of RNA called ribosomal RNA, or rRNA. So this is a third type of RNA that we're talking about today. So ribosomal RNA, or rRNA, is a type of RNA that's used to build the ribosomes. Okay. So here's a model of a ribosome. Ribosome has two subunits, a large subunit and a small subunit. And basically what's going to happen is an mRNA molecule is going to feed through this ribosome. And recall that mRNA molecule is 3-letter-long words, codons, that code for particular amino acids. And then in the mRNA, there's 3 different places where tRNA molecules can enter into the ribosome. And those are called the EP and A sites. Okay, so for example, the A site, what happens is a tRNA molecule, which has an anticodon that matches up with the codon on the mRNA. That tRNA molecule is going to enter into the ribosome, and it's going to bring its amino acid. So A actually stands for amino acid. That's where you're going to bring the new tRNA. That's where the tRNA is going to enter into the ribosome, bring in its amino acid. Okay, whereas the P site, P stands for polypeptide. That's where you have the tRNA that has the growing polypeptide, the protein you're trying to build. So P stands for polypeptide. Okay, E stands for exit. That's where the ribosome is, that's where the tRNA is going to go to exit the ribosome because it's already given away its amino acid. It doesn't have an amino acid there anymore. Or a little prettier version of this. So the A site is where a tRNA is going to enter, bring in its new amino acid. P site is where you have a tRNA with a growing polypeptide. And E site is where the tRNA is going to exit the ribosome because it gave away its amino acid. Okay, and we'll learn in detail how this works. Right now, all I'm trying to get you to learn is what the A, P, and E site stands for. A, amino acid, P, polypeptide, E, exit. Okay. So, in order to build a polypeptide, it happens in 3 stages, which are called initiation, elongation, and termination. So these are the 3 stages of translation. Now, of note, a little warning, like when you're reading the chapter, when you're studying for the exam, remember I didn't make you learn all the details about transcription. Well, transcription also has 3 stages, and those 3 stages have the exact same names. They're called initiation, elongation, and termination. So make sure you do not mix up initiation of transcription and initiation of translation. They're not the same thing. Elongation of transcription and elongation of translation are not the same thing. Termination of transcription, termination of translation are not the same thing. So we're not learning for this class the details of initiation, elongation, and termination of transcription. We are learning the details of initiation, elongation, and termination of translation. Okay, so just be careful you don't mix those up. All right, so to start explaining those, what I'm going to do is I'm going to start with a little animation.
In translation, a cell reads an mRNA message and assembles a polypeptide accordingly. We can divide translation, the process of building a polypeptide, into 3 stages: initiation, elongation, and termination. Translation is initiated when the small ribosomal subunit binds to the leader at the 5' end of the mRNA molecule. The anti-codon of the initiator tRNA binds to the start codon, AUG. The initiator tRNA always bears the amino acid methionine. Proteins called initiation factors help bring the mRNA, the initiator tRNA, and the small ribosomal subunit together with the large ribosomal subunit to form an initiation complex. The initiator tRNA sits on the P site on the ribosome. Elongation of the polypeptide now occurs, with amino acids being added one by one. The codon in the A site of the ribosome pairs with the anticodon of the appropriate tRNA molecule. Part of the ribosome catalyzes the formation of a peptide bond between the amino acid extending from the P site and the amino acid extending from the A site. The tRNA in the A site now translocates to the P site. The tRNA that was in the P site moves to the E site and exits from the ribosome. Meanwhile, a new tRNA brings its amino acid to the A site and the process is repeated. Three special base triplets, UAA, UAG, and UGA, do not code for amino acids, but instead act as stop codons, which terminate the process of translation. A protein called a release factor binds to the stop codon in the A site. It acts to free the completed polypeptide from the tRNA that is in the P site, and the translation assembly comes apart. The polypeptide becomes folded, modified, or combined with other polypeptides to form a functional protein.
Okay, now let me go through the 3 stages of translation a little more slowly. So the first stage is called initiation. Essentially what happens during initiation is you put all of the pieces together. So the pieces or the components that you directly need to be able to do translation is the mRNA, the tRNA, the amino acids, the ribosome, okay, and GTP, which is a source of energy. So those are the components or the pieces that you directly need to be able to do translation. An example of something that you do not directly need to be able to do translation would be DNA. DNA is not used for translation, it's used for transcription. Okay, so what's going to happen is we're just going to put the pieces together during initiation, the first stage. So what happens is the first tRNA molecule is going to bind to the mRNA. So the anticodon on the tRNA is going to bind to the codon on the mRNA. Now, the first tRNA molecule that binds, the codon it's going to bind to is the start codon. Remember, that's AUG. So this tRNA has an anticodon, CAU. Now, recall the start codon AUG also codes for an amino acid, so methionine is attached to this tRNA molecule. Then the two halves of the ribosome, the large subunit and small subunit, come together. So you basically just put all of the pieces together. And that's the initiation step. Now, when it's put together, that first tRNA is in the P site. What that means is there's now the next codon exposed in the A site. So the next tRNA that has an anticodon that matches up with this codon is going to enter into the A site, and that starts the elongation step. So this tRNA that has an anticodon that matches up with the codon in the A site enters into the A site, bringing its amino acid. Okay. Then what happens is the polypeptide that you've been growing that's attached to the tRNA in the P site, it transfers over onto the tRNA in the A site. So by doing this, what happens is we grow our polypeptide by one more amino acid. It was 4 amino acids long. Okay, now it's 5 amino acids long. And now the tRNA in the P site gave away its amino acids, so it doesn't have any amino acids anymore. And then what happens is everything just slides down one spot. So the tRNA in the P site moves into the E site, tRNA in the A site moves into the P site. Okay, so everything slides down one spot, and that tRNA in the E site then exits the ribosome because it's been used. It gave away its amino acid, so you're done using it to build this polypeptide. But then that exposes the next codon in the A site. So then another tRNA would enter, bringing its amino acid. Okay, and this keeps going over and over again, adding one amino acid at a time. Growing this polypeptide longer. That's called the elongation step. And this continues until a stop codon enters the A site. Once a stop codon enters the A site, then you start the termination step. So when the stop codon enters the A site, okay, what happens is something called a release factor enters into the A site. Remember, stop codon says stop translation here, and it also does not code for any amino acids. And this release factor basically just causes everything to be released. Okay, the polypeptide is released from the tRNA. The tRNA leaves the ribosome. The two halves of the ribosome fall apart. So basically what happens is you're terminating translation. You finish building the protein you were trying to build. You're done building it. Of note, these pieces can be reused, like the ribosome can be reused, the mRNA can be reused, even the tRNA can be reused. You just need to put a new amino acid back on it. Okay, so that's the third step, termination. So translation happens through 3 stages: initiation, elongation, and termination. Okay, so since we're talking about the process of transcription and translation, which overall is how you take the information that's in DNA and use that information to copy over into mRNA, and then use the mRNA information to build a protein. Something else we should discuss is, well, what happens when changes are made to that DNA? Okay, a change to the DNA is called a mutation. Okay, so what a mutation is a change to the DNA. And there are different types of mutations. We talked about very big mutations in a prior chapter, things like inversions and translocations. That's where you have big changes to chunks of the chromosome. Okay, well, there are also small mutations that can happen to the DNA. Those are called point mutations. A point mutation is a small change to the DNA, like where you change at one point, one little point, like one base pair, for example. Okay, so we're going to talk about what happens when you have point mutations. So here's an example. So we all have hemoglobin protein that we need to make. Hemoglobin is inside your red blood cells. It allows you to transport oxygen throughout your body. How do we know how to build that hemoglobin protein? Well, we have a gene that codes for that hemoglobin polypeptide. We copy that DNA over into mRNA, and that's transcription. Then we do translation to take the information in the mRNA and build the polypeptide or protein, in this case, hemoglobin. So we all have this gene. Okay, well, some of us have a mutation to the gene, a point mutation, a small change, where this T here has been turned into an A. Okay, well, then that changes the codon from GAA to GUA. Well, GAA coded for glutamine, but GUA now codes for valine. So this is a very small change. One single base pair has been changed in this gene that might be, let's say 3,000 base pairs long. Okay, well, that one change changes one amino acid, which that causes sickle cell anemia. Okay, so a very small change can have a drastic effect on the organism. So that's an example. So point mutations are small changes. Okay, they can be divided into two categories. So different types of point mutations is you can have a substitution. That's where one base is turned into a different base, like a base T is turned into a base C, for example. Or you can have insertions or deletions. That's where you add or remove like one or two bases. Okay, so we're going to give you examples of what these are and what effects they can have on the cell. Now let me explain in figure 4. So here's an example. So we have a piece of DNA, which would then code for this mRNA and code for this polypeptide or protein. Now let's say that we have a substitution. Okay, let me start over. Let's say we have a point mutation, which is a small change, and let's say that point mutation is a substitution. So this G is turned into an A. Okay, well that G was part of the codon GGC, which coded for the amino acid glycine. This A is now part of the codon GGU, which also codes for the amino acid glycine. Remember, the genetic code is redundant, so sometimes you can have a change in the DNA and the protein will be exactly the same. There is no change to the protein here, even though there is a mutation, a change to the DNA. This is called a silent mutation. A silent mutation is when there is a change to the DNA, but there is no change to the protein. Okay, another example. Again, a point mutation, a small change. Again, a substitution. So this time we take this base C and we turn it into a T. Okay, well, C was part of the codon GGC, which coded for glycine. T is now part of the codon AGC, which codes for a different amino acid, serine. Okay, so one amino acid has been changed. Glycine has been turned into serine. That's called a missense mutation. Missense is where one amino acid has been turned into a different amino acid. Another example. Okay, again, a point mutation, a small change. Again, a substitution. Let's say this T is turned into an A. Okay, well, T was part of the codon AAG, which coded for lysine. A is now part of the codon UAG, which is a stop codon. What that means, remember, stop says stop translation here. So instead of making lysine, you don't make that amino acid or any of the amino acids after that. Okay, so what a stop codon does is it stops translation. Okay, so when you create an early stop codon, that's called a nonsense mutation. A nonsense mutation is when you create an early stop codon. Now, out of the point mutations that I've talked about so far, the nonsense mutation that creates an early stop codon, is the one that's most likely to cause a negative effect for your cells, especially if it happens near the beginning of the gene. The beginning of the gene is called the promoter sequence. If this point mutation is near the beginning of the gene, well, the whole rest of the protein is not going to be there anymore. So that protein is definitely not going to work. Okay. Now, the other type of mutation, the other type of point mutations are called insertions and deletions. Insertions and deletions is where you add or remove like one or two base pairs. Okay, now remember how you read your mRNA. You read it within the reading frame. Like, for example, the example I gave you earlier was, you have these letters here. Okay, and then how you read is you read the first three letters, and then the next three, and the next three. So you read dog, ate, cat. Okay, well, let's say that there is a point mutation, which is a deletion. Okay, so let's say we delete this A here. Okay, so now the letters we have. Okay, so you still read the word dog, but then you read tech at, and whatever else comes after that. Okay, so what happens is we've just shifted the reading frame. Even though the letters CAT are still there, you can't read the word cat because you shifted the reading frame. This C is now part of this word. So what that means is every word after that is going to be the wrong word. That is called a frame shift mutation. Okay, you shifted the reading frame. Okay, and that can happen by if you get one base pair that's being deleted. Let's this example here. Okay, let's do another example. So again, let's say we start with this. So you would read it, dog ate cat. But let's say we have an insertion. Let's say we add a base here. Like let's say we add another G here. So now we would read dog still, but now we read gat, eka, and whatever else would come after that. So even though the letters ATE and CAT are still there, you can't read the words ate cat. Because again, you shifted the reading frame. Okay, so that's called a frame shift mutation. Okay, a frame shift mutation happens when you add or remove like one or two base pairs. That causes a frame shift mutation. So here's an example of that. So we have a point mutation, a small change. This time the small change is a deletion, so we delete this base here. Okay, so it's gone. That base A was part of the codon UUU, which coded for phenylalanine. But now this U is gone. So instead you have UUG, which codes for leucine. Okay, and then instead of GGC, you now have GGU, which codes for alanine. Okay, so what happens is every amino acid after that one base pair deletion is going to be the wrong amino acid. Okay, so that's a frame shift. Now, this is another example of something that's most likely to have a very negative effect for your cell. If you add or remove like one or two bases near the beginning of the gene, near the promoter sequence, then the whole rest of the protein is going to be wrong. Okay, so that's very likely to have a very negative effect. Now, if you add or remove three bases, that will not cause a frame shift. Like, let's say we have these three bases here, TTC, and we delete them. Okay, so if we delete them, then we get rid of this codon AUG, we get rid of this amino acid lysine. So in that case, we don't cause a frame shift. Okay, we still read phenylalanine, glycine in the same way. We're just missing one amino acid. So a frame shift is when you have an insertion or a deletion of one or two base pairs. If you insert or delete three base pairs, it doesn't cause a frame shift. So those are mutations. Well, how do we get mutations? Some mutations are spontaneous. They just happen all on their own. There's nothing you can do about it. For example, during DNA replication, what we learned about in the last chapter. Okay, DNA polymerase III does a very good job of copying your DNA, but it's not perfect. Once in a while, it makes a mistake. Like, let's say it accidentally puts a T instead of a C. Okay, well, that creates a substitution. Or let's say it accidentally forgets to add a base. That's a deletion. Or it accidentally adds an extra base. That's an insertion. So while DNA polymerase is very good at copying your DNA, it's not perfect. Once in a while it makes these mistakes, and they just happen naturally. Okay, so some mutations are spontaneous. They happen all on their own. Some mutations are because you're exposed to mutagens. Mutagens are things that will mutate your DNA. For example, cigarette smoke. Okay, if you are smoking 2 packs of cigarettes a day, then it's very likely that sometime in your life you're going to get lung cancer. Why? Because that cigarette smoke contains mutagens, okay, and those mutagens mutate the DNA of your lung cells, causing those lung cells to become cancer cells. Or another example is sunlight. Okay, it's not good to get too much sunlight exposure. Why? Because sunlight, what it is ultraviolet radiation, UV rays, UV radiation. Okay, which is a type of radiation. What radiation does is it mutates DNA. So that's why if you get too much sunlight UV exposure, it can mutate the DNA of your skin cells and it can lead to skin cancer. Okay, and so that's where you get these mutations. Some happen spontaneously all on their own. Some are because you're exposed to things like cigarette smoke or UV rays. Okay, And that is the end of chapter 17.